Awesome. Thank you, everyone. Thank you, Carlos, especially for the invitation. And thank you all. It's the end of the day. I'm sure I saw the schedule, and it's like you guys covered so much already, and thank you for coming back. Um, yeah, I'm excited to share some of the new work that we've done in my lab at UC Riverside. So I'm a very young PI, so I've been there, I think, a little over a year and a half. So it's like nothing, but there's some new projects that we're excited to share, and we'll kind of go from there. And if you have the Google Drive, I just uploaded the talk to there. Feel free to download it. As part of this, it's going to have a few interactive portions interspersed. So I'm trying a new thing. So let's see how it all goes. Okay, so broadly, here's my disclosure, number one. And so broadly, just to give you an idea, a high-level idea of the pieces of the ecosystem of GNPS1, and we'll talk about GNPS2 in a second. But kind of the ways that I think about this, there's one portion, there's algorithms, visualizations. These are the kind of tools that take some data to do something. We also have the actual data itself. So there's some public data, or it can be some private institutional data. And this is something that we want to work with, we want to mine, we want to interact with it. It could be your data as well, right? So this is kind of this big mass of stuff that's just kind of we have to store. As well as we want to be able to distill down knowledge bases and collect knowledge from the community that we can reuse in the algorithms to analyze the big data. And so in some senses, the knowledge bases is a very big subset or reasonable subset of the big data that has been curated by scientists and people and carefully managed into something that's a little bit more useful than kind of the raw stuff that's coming off the mass spectrometer. So those are kind of the three pillars that in my mind, that make this all work. And so just kind of give you an idea over the years, we started from very humble beginnings back in 2014. I still remember like kind of the very first things we did, building the GNPS platform, building the tools. And I think it's so funny. It was Christmas break, like the holiday break in 2013, where I didn't go home. I just didn't want to go home that year. And I spent the whole break kind of working on this pet project of mine. And we still use some of that code for, I wouldn't say for better or for worse. It's like for the worst, we should probably replace it. But it's very funny how something you do at this point 10 years ago, and it's still, it's still very cool, but as a joke, you're still paying for it, right? So you made some mistakes, and anyway, it's just very interesting to think about. But over the years, this plot here on the x-axis is time, and the y-axis is the number of analysis jobs that are run at GNPS. And so I stopped measuring this back in 2023 when I left UCSD. But we can see like this large growth. In total, it's about 600 to 700,000 analysis jobs. And when I last measured it in 2022, it's the system and the users, they'd analyzed about 25 million LCMS files. So it's not a small amount, of data. And I just think it's very cool, and we're very proud and kind of just very thankful that the community even gave us a chance to just use our tools. So that's kind of cool. So kind of some new updates in the last like 9 months. Just kind of setting the stages. There's a new arrival to the lab at UC Riverside. So about 8 or 9 months ago, I can't even keep track anymore. My son was born. So it was my first child. And one of the problems with children, okay, maybe not a problem, is unlike a computer, they don't stop doing their thing, and so they have a constant demand on your time and attention. And so they're lovely, they're great, but one of the things is keeping a web service and a web tool available for the community 24/7 up and running is not easy, right? So there's systems. It's just a big system that things break. These are physical systems that they break, and so what kind of the problem that arose, especially in the first couple months when my son was born, is back in July we suffered several days of downtime. But I got a baby at home, so what am I going to do? What are you going to choose? And you're generally going to choose the baby. And so this is not good for the community, and it's not good for us because we want to provide service. So we want people to use our tools. We want to move science forward in the way that we can, and then that way is we can make tools and we can make them easy to run for you all to use. And we get, number one, I get dozens of emails when anything goes wrong. But it's a disruptive analysis work. And so this kind of crystallizes for us that there's a definite need for resiliency. And so we would love to provide a service for everybody, but we also understand that, especially, I've been on call effectively for the last 10 years, since 2014, to make GNPS work and run all the time. I mean, sometimes we're successful, sometimes we're not, but that constant maintenance, I think, has paid off in the long run. But it's just not, it's hard to do once you have kids and all these other demands as a PI. I'm just kind of recognizing that, yeah, we want to have build resiliency, and then we're going to tell you a little bit about GNPS2 and how we achieve that, or how we aim to achieve that. And so one of the things when we originally started GNPS was it was all housed at UC San Diego. So I was a grad student and a postdoc there, but I left. I left, and I'm no longer at UCSD. And so the question is, how do we start having resiliency? How do we provide these tools when I'm no longer at the institution where GNPS lives? And GNPS as an infrastructure can only be in one location. That's just the way it's been designed, and it's been engineered. And so the idea is we can keep a lot of the things that are difficult to move, the big data and the knowledge bases that people rely on. We can keep that at GNPS1. We can keep it at UCSD. And those are relatively, at least the knowledge bases especially, are relatively easy to keep up and running. The hard things are these algorithms and visualizations that always have to work. And so these have been migrated over to GNPS2 at UC Riverside. And so we've developed a new platform, and we've refactored a lot of tools, developed some new tools, but we'll go through how this enhances resiliency in one part, or several different ways, for you all. And so first of all, we can deploy GNPS2 on any institution on commodity hardware. So you just have to have a CPU server, you don't need GPUs and things like that. You can kind of deploy it anywhere. So we can start thinking about not just at UC Riverside, but for a wider deployment base. So then you can go to different places, and you can access the same services. So you wonder how Netflix, is resilient and Google's resilient. They do this, but at a much larger scale. So we want this to be flexible as well. So not only can you run the workflows at GNPS2 at UC Riverside, you can run it at GNPS2 at other sites. And if any of these web services are down, you can run it on your own computer. So that's kind of a change from what we did in GNPS1. Now we can actually run it outside. We put a significant amount of work to make that happen. And also, it's easier to develop workflows. Before, we required privileged accounts. Basically, you had to be at UCSD and an employee of UCSD in order to do it. But now anybody can develop workflows. If you have a tool that you want to put into GNPS2, we have a whole development ecosystem, and it's all built on Nextflow. And if you want to deploy and see your tool in action at GNPS2, let us know. It's super easy. And there's this aspect of shareability. And so you can access data from almost anywhere. So GNPS, metabolites, metabolic workbench, if you're doing proteomics like PRIDE. And if you want to reanalyze public data, you can source those from anywhere and analyze them in GNPS2 pretty seamlessly. But also, you can share and synchronize your analyses from GNPS2 with others, and synchronize to other sites. So you can deposit the Zenodo, and you can do things like that. So it makes sharing your analysis that you do at GNPS2 a lot easier. Again, it provides resiliency. So if you need to access old data or visualizations, and GNPS2 is down, you still have that data and it's yours. So it's not really locked into an ecosystem. And finally, one of the things that we're starting to push with deploying new GNPS2 instances around the world is privacy. And so, it's running at UC Riverside right now, the main one. But sometimes your data, especially if it's clinical, cannot leave the country. And so we're bringing these, or at least the continent. So having resources in the EU, having them on each continent can help manage privacy. So, but you can use the tools without having to surrender or trust that we won't look at the data. Not that we will, because we're so busy. Why are we looking at your data? But it's more of a kind of funding mandate and kind of restrictions within your institution or within the project. So probably a pretty good idea to adhere to those things. So those are kind of the things, the key problems that we wanted to address with GNPS2 over GNPS1, and these are becoming more salient, especially in a time of GDPR and things like that. And so it's live today. So I think we've actually, you've played around with it yesterday and a little bit today in some of the workshops. So you have accounts, you're familiar with it. I don't need to talk about this, but just to give you an idea of where we're standing right now. So we have GNPS2.org up and running at UC Riverside. And these are running on my lab servers. And it's a very modest setup, but I think it's at least modest by high performance computing standards. But relative to the average desktop, it's kind of an elaborate setup. But we also have a version running in Germany. So Daniel Petrus's group, who was formerly at Tübingen, and it's running on the DenB cloud. So that's up and running at de.gnps2.org. We're building some new infrastructure so you can start using this hopefully soon. And we are in the works of bringing something up in Brazil. So at least we're spanning several continents, and we're figuring out a few more as well. So that's kind of the state of the art here. And so one of the things that I want to talk about today is some of the newer, so those are the kind of infrastructure, how do we make it, that's always available. And, I think it's very exciting. But for you all you care about is when you go to gnps2.org, it loads and everything works. So, the Google, Facebook, all of them have ruined things for all of us. They've set the expectation bar for web tools so high that you all have an expectation that everything works. That's good. It's good. But it's just very interesting to me that I find it interesting. It's not an easy problem from the technical side, but the expectation from users is that, well, of course it works 99.99% of the time. So, so we'll move on and talk about the more exciting parts and some of the newer tools to analyze data. And so one of the key things is we have a lot of data available at in the public space, and we have some algorithms. So how do we put these two ideas together and enable utility from this big data? So that's kind of part of this talk will be today. And so one of the questions out there is, how much data do we actually have that we start mining and making use? And so there's, at least at GNPS, the public data sets, there's about 60 terabytes of public data. And this is about 200,000, 250,000 LCMS runs. So it's pretty big. I mean, it's probably more data than any of you individually have ever generated. So that's kind of exciting. And it's growing. And when we consider all public repositories, we're talking about over 1 million LCMS runs. And so that's kind of the space that we're operating in. And so one of the parallels that we want to make to this is, in the sequencing space, one of the things that is readily accessible and readily you can do is you can take a DNA sequence and ask, has this DNA sequence or any structural analog or any analogs with polymorphisms, for example, occur in any of the public data? So this has kind of been par for the course for the last 20 years in sequencing at NCBI and their BLAST set of tools. And so you're able to mine a huge and leverage this huge amount of sequencing data and be able to ask with a simple query. And so what we want to be able to do here is create a new tool for the small molecule mass spectrometry space that does the same thing. Instead of taking a DNA sequence and blasting it against all SRA or all of the NCBI taxonomies, all the assembled genomes, we want to take tandem mass spectrometry, which is to us kind of a fingerprint for a molecule, and ask the question, has this spectrum that represents a molecule ever been seen before in any of the public data? And has any analog been seen before in any of this public data? And again, just to show you the scale of where we can start doing the search, thousands, over 2,000 data sets, about 1 billion tandem mass spectra that we can start searching. And this is from mass spectrometry-based metabolomics and natural product data sets predominantly. So it depends on your question and the space that you're working in, but the public data covers kind of a wide variety of resources. So just to give you kind of one example of why this is particularly useful. So imagine, there was a study like 5 or 6 years ago that we're doing at UCSD, and they were looking at non-alcoholic fatty liver disease, and they considered the unhealthy patients versus some healthy control. And then you're looking through this untargeted data, you have thousands and thousands of features, right? And there was one feature that popped out that seemed to be higher and elevated in the unhealthy patients compared to the healthy controls. So that's where we start. You can do the typical thing, you can start solving the structure and things like that. But one of the things that we can also do is we can ask the question, how relevant is this particular molecule? And we don't have the full structure because we only have the tandem mass spectrometry data. Where else have we seen this molecule, and is it relevant in kind of a healthy versus disease kind of condition, so that we can think about, is this kind of a spurious statistical artifact, or is this something that's meaningful for us to follow up on and actually work to solve, purify and solve the structure? And so we use this mass spectrometry search tool, so we call it MAST, and we search against all this public data, and we found, hey, it was observed in 43 data sets that have the exact same MSMS data. And so when we look deeper at some of these data sets, we could find that not only was it found in other data sets, but it was found in some mouse data sets that were studying their stool, and we found that there was a high-fat diet versus a normal control. So it kind of simulates some of the issues that come about with non-alcoholic fatty liver disease. And so this particular metabolite was higher in a high-fat diet versus a normal control. And so that gave us a little bit more confidence that, oh, this metabolite has kind of an interesting and similar pattern in other biological contexts. So this is something worth following up on and kind of putting more work into investigating the biological role of this particular molecule. So that was kind of the one small vignette on contextualizing the role of a metabolite before you actually can identify it by using all this public data. So hopefully that sparks a little bit of imagination on what you might be able to use it for. Other things that other collaborators from the natural product space, so we're talking about eco-metabolomics here. So if you're talking about from the natural product space, maybe you have some microbe that's producing some metabolite, and you can ask what other microbes might produce it, and what other ecosystems might it be observed. Is it like in the ocean and things like that? So you can use MAST for that. One of my collaborators, Vanessa Phelan, who's pretty clever, and she's a natural product chemist. And so what she uses MAST for is she likes to discover new molecules. And whenever she discovers a new molecule in the mass spec in one of her microbial isolates, she typically needs to purify it and then and do NMR to solve the structure. But the problem is sometimes your bacteria, they just don't produce very much of that metabolite. And So she was clever. She's like, well, I'm just going to use MassQL to find a more prolific producer, so it makes my life a whole lot easier. So then she can find another microbe that actually produces the same molecule at higher titers, grow that one up, and then it's so much easier to get enough material to solve the structure. So it's a kind of a improves the operation of your lab. So that's one thing that we think MassQL can be useful for, or several ways that MassQL can be useful for. But one of the things that we published this paper back in 2020, the original paper back in 2020, where you can take an MSMS and you search all of this public data. Now, we have high expectations, and so the original problem was that it works, but it's slow, right? And so a single query would take about 15 minutes. And then once you do a workshop like this, it becomes way less fun. And so one of the things, there's a little pun here. So it's like, if Mass is slow, we can make it faster. So anyway, one of the things that we did recently, we published a few papers around this, is how do we start accelerating Mass from minutes across all this big data to milliseconds. So we're talking about several orders of magnitude. And so the key insight here, we won't go into the details. The key insight here is that the spectra are actually very sparse. There's not peaks everywhere. And so if there's not peaks everywhere, you don't really need to look at the places where there are no peaks. And that's what we were doing before. And you load up all this data that doesn't actually even match to your query. So it's like, why are we even looking here? And so we leverage this sparsity idea of peaks not occurring at all masses. And then we can drastically look at subsets of the data, even when we're talking about 50 terabytes of data. And what that results in is about a 3,000 x speed up. And so we translate from 15 minutes to a few milliseconds. So that's where we're at today, and that's where all the tooling exists today. And so we're going to do a little quick hands-on. And so this is actually a new tool that's built on top of MassQL. It's called MicroMast. So we just published this a few weeks ago, somewhere, Nature Microbiology, I think. And it combines this idea of searching with extensive microbial and taxonomic metadata. So we know what sample comes from which microbial isolate. So you have your computers in front of you. Go ahead, and you can download the slides as well. But you can go ahead and click on either this URL, or you can type it in. But it's probably easier just to click on it. So that's what I am going to do. Or actually, I'm just going to go to right there. And so you'll see something like this if you go to the website. And once you're there, what we're going to start trying to do is we're going to search for, we're trying to ask what microbes produce a particular molecule. In this particular case, the example molecule is lovastatin. And so this is a drug now that's being used to treat high cholesterol, but it originally was a microbial natural product. If you go to Wikipedia, they'll tell you who makes it. But we want to ask here purely from the data, can we tell what is the producing strain of lovastatin? Here's the structure, and just kind of just to show you, here is the tandem mass spectra, just kind of a picture of it. But once you're at this particular website, there's a Spectrum USI. So this is the Universal Spectrum Identifier. This is basically an address to refer to a tandem mass spectra somewhere in the cloud. And this is the address for that particular tandem mass spectra for lovastatin. And so if you paste that in there, and yeah, so that's the very first thing. And you click the button Search MicroMast by USI. Then we can actually figure out what type of bacteria, what type of taxonomy actually produces lovastatin by doing the mass search and then combining with the metadata. So just to, you'll have this slide here, but just to remind you, this is the website. There's actually an example here. If you don't want to enter that, you can click on lovastatin here, and that will pre-populate this just as a little shortcut. So we'll give you a few seconds to do that. And Carlos will let me know if there's any questions. So if you do have anything, flag down Carlos. You can use the microphone, or you can type it as well. So, and if you have a quiz, if you the camera is actually on the audience. So if you do have a question, or it's like going very badly, feel free to just kind of wave Mayday or something like that. So. Okay, were you all able to get to there and try to enter lovastatin there? Yes? Okay, I see one thumbs up. Awesome, awesome. Okay, so what you'll notice at the very bottom of this page is that there's a little tree. Okay, this is not really, this is technically a tree. But it'll tell you the hits to microbial metabolomics data. And because we have the metadata and the full taxonomy, we can visualize a lot of this as a tree, and you can figure out which clades of bacteria or microbes have we observe this particular metabolite. And so if you do that, this is what you'll see. So at the very left, it's the higher levels of the taxonomic tree. So it's like eukaryote, fungi, and then all the way to the right, if you go all the way to the right, it'll tell you it's an aspergillus, species. And so this is actually what the actual producer that's well known for this particular natural product. So this is one example of, you can take your data, search against all this data, combine it with the microbial metadata, and we'll hopefully tell you the breadth of the taxonomic space where we observe this particular metabolite. And so you can do this with any tandem aspect in your own data. So hopefully that's useful and interesting sometime in the future, as one way that you can start leveraging all of this available public data. Does that make sense? Any questions? Okay, so that's MAST, and definitely play around. Freely available, no logins required. And if you want to do this on a really large scale, there are workflows for all of this that you can hit the APIs and do hundreds of thousands of tandem aspect. Should be no problem. You just might end up with a giant set of results that you'll have to sort through then. But it's, that's maybe that's the fun of it. So the next thing that I want to talk about is a little vignette about mining for specific compounds in public data. And so, working with Peter at UCSD, we have a very good working relationship because he's a mass spectrometrist, a chemist, and I'm a computer scientist. And so we have different expertises, and we have different capabilities and different perspectives on what we can do. And so we're just good friends, number one. And it's very fun that whenever we both work together, we come up with new ideas and new ways that we can build, and they can kind of exploit these discoveries. And so just kind of chatting with Peter, one of the things that he idly asked about one day a few years ago, and it was, well, we have all this public data. Can we just find all carnitines in public data sets? And so kind of my thought process, I know a little bit about chemistry, is that I know certain molecules imprint certain data patterns in the mass spectrometry. So for example, here, if you have certain atoms, they imprint certain isotopic patterns. If you have certain atoms, they, like on the left-hand side in MS1, you have iron has a particular pattern. Here in the second column, bromine has a very distinctive isotopic pattern. Sometimes whenever you have a particular structure, it produces unique peaks or losses in the tandem mass spectrometry data. So that's something I knew, we know that happens. And we know some rules about fragmentation, not all of them, but some of them that are enough to be useful. And for carnitine, so I know, okay, it has this kind of quaternary amine, and it likes to have all the charge. And I happen to know that there's like these 60 M over Z and 85 Dalton peaks that are interesting, that are always observed with carnitine. They're reasonably always observed in carnitines. And so I thought to myself, well, I can write a little piece of software that searches exactly for carnitines, right? And then we can deploy this on all this public data, and then we can answer this question. Well, the problem is, once, there's a saying in English, at least, like once you give a mouse a cookie, they'll want a glass of milk, right? And so once you can do it once, they'll want to do it on everything, like carnitines, brominated compounds, siderophores, saccharides, some peptide natural products, bile acids, all this stuff. And there's no way, like one of the intrinsic qualities of a computer scientist is we're really lazy. We don't really like to do anything. And so we're not doing this. Like, we're not going to custom write a piece of software for everything that a chemist wants to do. And so kind of the thing that kind of was asked is, can I develop a kind of a universal computational solution so that chemists can do this themselves, so we don't have to actually do it with the amount of expressivity that they actually want? And so we think the answer is yes. But the way that we achieve it is by creating a new language, and it's called the mass spec query language, or MassQL. And so kind of just walking through this, here are the several pillars and kind of the design philosophy that we went through. So first, we want MassQL to be understandable. So it needs to be easy to read and write for a chemist. If you're going to try to design all these data patterns, well, we want it to be super, super easy for chemists. Number two, it needs to be flexible. So it needs to describe every sort of pattern that you would actually want to find in mass spectrometry and mass spectrometry adjacent. So retention time and ion mobility, kind of the intersection of all three of these things. It needs to be scalable. And so not only do you want to be able to query your data, but you probably also want to query all public mass spectrometry data out there. So we're talking about, again, billions of tandem mass spec. And it needs to be reusable. If you're going to take the work to codify something, maybe you should be able to give it to a lab mate. And then it's reproducible, and it's unambiguous, and it's just a way to store knowledge. So just kind of going through each of these facets to make it easy to read and write for chemists. First, we want to, if you have a query that exists, we have these MassQL queries that we have examples. To explain it to people, we can translate it into English here. It's kind of a auto machine translation. It's not very good, but you can kind of get the idea across, and you can kind of see there's a little bit of syntax for MassQL, but it's kind of near English, and you can kind of piece together the semantics of MassQL just by looking at it a few times. But since the time we invented MassQL, some things have changed in this world. So one of the big things is these large language models have become significantly better. And so one of the things that we can do now is not only can we translate from MassQL into English to explain it, kind of in an automated way, but we can go the other direction. We can go from some sort of natural language, whatever you all want to type. If you don't know MassQL, and it can go into MassQL. So for your first foray, you don't even need to know the syntax of the language. We have a little large language model. It's called a MassQL assistant, that we tell it all about MassQL. We tell it about the syntax and all that stuff. And you can chat with it. So that's one of the things that we will do interactively in here. So we're pretty excited about that. Also, to make it, actually, it's very interesting. It does a better job of explaining MassQL than our own software of explaining MassQL. It's kind of embarrassing, but it's actually very cool. So another facet that I wanted to mention is that it's flexible. So we think you can describe all these complex mass spectrometry data patterns that you would want to start expressing. And so just very quickly, this is kind of the skinning the surface level. But at the top left, if there are certain peaks that you want to find, you can express it. I want to find a 297 peak, for example. You can start looking for neutral losses for the precursor. And then down here, you can start looking for neutral losses from any peak. So basically, I view them as peak gaps, but you can also term it as neutral losses. And also, you can think about relative peak intensities between several peaks in the tandem mass spectrum. So these are kind of the most basic primitives that you can use to query for tandem mass spec or MS1 data, or a combination of all of them. And you can combine multiple of these literals together, or these primitives together. So you can say, I want to look for a peak and a neutral loss from precursors. So you can start having conjunctions and really putting together a complex query to look for what you want to find. So just to show one example, so you all met Daniel, and so we were just kind of in the early days playing around with this. And here's one example that he put together. Is during his PhD, he was looking at a class of natural products called aldecidins, and he knew, because he knows things about chemistry and biology, that there's a 468 and 660 peak that are consistent for this family of natural products, these aldecidins. And so he was able to write a query very quickly here. And then what he was able to do was deploy this query in an afternoon on his old mass spectrometry data that he had published a paper on back in 2017. Now, what he was able to do in this paper, or in this data, he was able to very quickly rediscover the albicide molecules in this kind of in a LCMS data set, all the molecules that he published on originally back in 2017. So yeah, easy, quick, straightforward. What took him probably months originally, you can do in a few minutes. But what's more interesting here, and this is a molecular network showing his data, is that he was able to putatively identify more members of being albicidins in the data than he had originally discovered in his paper. And so each of these circles, each of these nodes is a tandem mass spectrum, and in purple or in blue are the known albicidins that Daniel published on, and in gray are the ones that he didn't publish on, right? That MassQL actually found in the data. And so MassQL and visualized with molecular networking revealed new putative analogs that are candidates for structural elucidation. And the reason that they weren't found is because you only have finite time to look through data as a person. But with MassQL, it finds exactly what you're looking for in an exhaustive way. So it's that exhaustive feature that scales your effort that allows you to be comprehensive in discovery. So anyway, it helps generate new hypotheses that you just couldn't have reached before. So we thought that was pretty cool. And this third pillar is that things are scalable. So we want to really get to the repository scale and see what uniquely we can find in the repository scale. And I didn't have time to include this here just because on Monday we actually published a paper in Cell that really does the kind of really first published example of a repository scale query that discovered thousands of new bile acids that were previously unreported. So that was pretty cool. But we're going to take you through an example in this particular talk that's slightly different. And this is a work by Nina Zhao. She's now a postdoc at UCSD, but she really cares about environmental pollution. And so there are classes of compounds called organophosphate esters. They're toxic to humans. They're inflame retardants, and they enter our waterways. So it's not great. We really want to know what what's out there. What is the family and diversity of organophosphate esters, because there's hundreds of analogs, so that we can start being able to quantify that kind of later. And so the very first question is, how do we start figuring out what is that diversity? And so one way that Nina went about this is that she knows that these organophosphate esters, they have this phosphate group that has a very distinctive MSMS fragment at 98.98 MOZ. And so she wrote a pretty quick MassQL query that said, I want to look for that peak, and it's got to be at least 50% of the intensity. So it has to be a pretty big peak in that spectrum. So what we're able to do is, because we built this whole ecosystem and infrastructure, is go to the 1.1 billion tandem mass spec data, apply this MassQL query, and find exactly what we're looking for. And so this whittled 1.1 billion down to about 338,000 tandem mass spectra, kind of several orders of magnitude filtration, something that you just could not do manually. Then we still wanted to kind of collapse this scale a little bit further. So we clustered identical MSMS spectra with Falcon. We ended up with about close to 3,000 unique tandem mass spectra. And then we did molecular networking at GNPS. And we had about 169 molecular families. And so now we can start organizing these together and start exploring this diversity for finding new molecules. And so here's one particular molecular network. Again, each circle here, each oval, is a unique tandem mass spectrum, and they're connected if we think they're structurally similar. And so this particular family happens to be the organophosphate esters. But what's interesting here is less than 10% of the organophosphate esters are of known masses, right? And so things in pink are the ones that are known. Things in blue are the ones that are unknown. And what Nina was able to do by mining this data after we did this repository scale search is she was able to find new organophosphate esters in her own published data. So just like Daniel, because MassQL was exhaustive, she was able to discover new molecules that she didn't find when she did the manual analysis of her data. So that was kind of cool. But more uniquely is, because we scale this up to the repository, we were able to find new organophosphate esters that came from other data sets that were not Nina's. And so by doing this repository scale search and using MassQL, we enriched for organophosphate esters, brought them all together from many, many data sets, and was able to prioritize, or visualize and prioritize and discover new organophosphate esters by combining all of these techniques together. So we thought that was pretty cool. Again, there's a more interesting, I think, biological story with the bile acids, but it takes a very similar tactic. It's this idea you have so much data, you cannot sift through it. You have some sort of hypothesis you want to find, enrich for it with MassQL, take that data and then visualize it and make some new discovery. So it makes billions of spectra down to thousands that you can actually prioritize and make a discovery. So I think that was pretty interesting. And the final pillar here is that whatever you write, whatever queries you write, they're reusable. And so one of the things we made is a compendium, so you can contribute MassQL queries that you've written and share it with the rest of the community. Or for kind of a purely selfishly, you can go there and see what other people are doing. So you can take their queries and quickly apply it on your data. So things like looking for sugars or looking for brominated compounds, for example. But one of the key things is probably sharing it within your own labs, because you guys are looking for working with specific compounds, types of compounds. And if you write something that's interesting and useful, well, give it to a lab mate. Or if you're a PI, you can give it to your student or a tech, and it's reproducible, and they're doing the right thing. And you can also check their work. If they've written a MassQL query, it's a little bit easier to make sure everything is done properly, rather than a kind of an oral description of what's being done. So we think that's kind of interesting. And finally, what MassQL is in the community, we have Python and R APIs for MassQL. So you can if you like programming, you can use it in a lot of your programming languages. So this is open source, and it's more or less free for industry and academics. And we've also integrated MassQL into community and commercial tools. So MZMine, MSDial, OpenMS, GNPS2, Unideck if you're doing top-down, and commercial tools such as Metaboscape from Bruker. And so the idea here is we have a universal language. So this is the same language in any of these tools. So no matter which tool that you want to use, historically you have to learn a new user interface every time you go to MZMine or OpenMS. It's a totally different interface. It's done a little bit differently. But with MassQL, if you write MassQL in one software, you can take it and do the exact same analysis in another piece of software. So if you're sharing it again with the community or collaborators, it's just a little bit easier to be reproducible across different software. So you can definitely try this today. And we're going to walk you through a few examples that we can do in GNPS2. So any questions? Any questions so far? Okay, we see a hand. Carlos, do you want to, if possible? I have no idea how big the room is. So
there's a question here.
I have a question about the Python API. That was what I was about to ask. Could you please talk a bit more about this, because that's still something that we want really to use it, and for many more people maybe that's a question like what would be like the file that we can use it, like the input file,
right
data, and what would be the Python package that we can use for that.
Right. So there's a MassQL package and it's open source. So you can use it in two ways. Number one, you can import it as a package, so then you can write it in your own software if you want to integrate it into like an existing piece of Python code. So basically you can say massql.query. So you can just say you can query an MZML file inside, like you can call it inline as part of an API call. We natively read MZML, MZXML, and MGF files. Alternatively, if you have your spectra, all your spectra in like data frames, like a pandas data frame, you can just pass it to, there's an API within there. It's like, do the query on this data frame that represents the mass spec data, and it'll do it. So those are several ways that you can use the package integrated into your own software. If you like Python notebooks or whatever, or Jupyter notebooks, you can do that. Alternatively, the Python API specifically, it also has command line entry points. So you can run it on the command line if you just want to say, here's a query, here's a file, give me an output TSV file. It does that as well. So there's multiple. It's a command line tool and kind of a package API that you can build into your own software. Additionally to that, we also have a Nextflow workflow. I don't know if you're familiar with Nextflow, but it's a command line Nextflow workflow that instead of being able to run it on a single file where you use the Python script on a single file, this allows you to point MassQL at an entire folder. So we're talking, the way we use it in GNPS2 is you can point it at like 150,000, a folder with 150,000 MZML files. It will do it in parallel, and then at the end of the day, it'll aggregate and give you a giant result file. And so if you want to scale, basically it's a question about scale, right? So you can do it in your software for doing it for one file interactively and all that stuff. Or if you want to use the Nextflow workflow that integrates like the command line tools and parallels and all that stuff, you can scale this up to, if you're a company, that's all of your internal data inside the company. Or if you're a lab, like all of your historic data that you've ever collected in your lab, if that's the scale that you actually want to apply it to. Does that answer your question?
Yeah, thanks.
Any other questions?
Any other questions? Yeah, we are good to go.
Okay, so let's have you play with a little with it. If you have not, ever worked with MassQL before, I think this will be fun. So the very first entry point is how to write your very first MassQL query. So in the slides, you go to this link, or you can just type it massql-analysis.gnps2.org. So I'll just show you what this sort of looks like. So you'll get to something like this. I'll keep the URL up here. Okay, was everybody able to get to the website? Okay, so once you're there, one of the things that you'll see, okay, so I gotta show you one thing. So there's a little password. We actually use OpenAI's API, so we actually pay for the access. So the password is MassQL, all lowercase, no spaces. So if you type that in here and hit enter, it'll grant you access here. So that it'll just be, yeah, MassQL. It'll look like that. No capitalization, no spacing. Okay, is everybody able to get to this a blank chat window now? Okay, so here we can start writing our first query. So just as an example of one thing that I pulled off, you can just say, write me a query to find MS spectrum with a peak at 59 and a neutral loss of 85. Or you can do some variant of that. So I'll just do it live here. Write me a query for MS spectra with a peak at 59 and a neutral loss of 85. And so this happens to be valid MassQL syntax. And it just kind of does it. So you can do fancier things beyond that. So one of the things you can do is say, whenever you want to look for peaks, I want them to have a certain tolerance, right? So can you add a tolerance of 50 ppm to all the conditions? And here we go. So it'll add a 50 ppm tolerance to the product peak and the neutral loss. So it's a please format each condition. So this is just a formatting thing, but you can have a conversation with it, right? And you can say add another condition of a peak at 112 m/z. So there we go, right? So and then you can take this query and you can use it in the MassQL ecosystem. But the idea is you're kind of learning MassQL kind of passively, but without explicitly learning it, and that seems like a lot of work. Is this all working for you guys? This is the first time we've done a workshop with this chatbot, honestly. So I'm just curious. Okay, that's great. That's great. But by the end of the day, we want to lower the barrier of entry for you all. You know, the funny thing is I invented the language for MassQL. So I wrote the formal grammar and everything. I actually use this now because I'm super lazy, because sometimes I don't even remember the syntax. So it's kind of interesting. And if you want to share kind of your chat history, just if you think it's interesting, or just to save it, you can click on link to chat history up here. And we save the chat link. So then you can kind of see how you achieve what you achieved as part of the chat history. So anyway, does that seem reasonable? Any questions? Okay, awesome.
There's a question here.
Oh, okay, yeah.
I have a question about where an example where you showed where you said something is y and y, and then another peak is y times 0.6 or something like that.
Oh, right.
Yeah. So that doesn't seem to be. Is there a group? I mean, does that imply greater or smaller? Because exactly 0.6 is of course not what we want.
Right. Right. Exactly. So that's a good question. So the way we do that, I mean, let's just show this example here. So. Right, so the intensity is y times 0.6. And so this implies it must be 60%, the second peak must be 60% of this first peak. But there's another condition, I've removed it here just for brevity, but there's another kind of qualifier there that tells you how much wiggle room there is. So if this first one is intensity Y, the second one is never going to be exactly, as you're saying, never exactly 60% the intensity, but you can say, but it can be within 25% up and down of that kind of expectation. So that's something you can tune. Does that make sense? So yeah, you can basically define error bars of the secondary peak that defines how much it can vary. And there's actually let me show you this. So I don't know if we have an example here, but there's a MassQL kind of like workspace here, and let me see if I can copy this, because we actually try to visualize the queries so that it does show you the error bars. So sorry, I don't have a good example right here, but okay, there's not a good example here. But follow up with me if you actually want to take a look at this. But people have done this. We've done this quite a bit we've done this quite a bit with looking at MS1 data, like isotopic distributions. And so we end up playing around with the tolerances quite a bit. And at least to my surprise, even things like bromination, where if you have a single bromine, they should have the same intensity. Peak intensities vary a lot more than I would have expected, but that's something you can't adjust. Any other questions? Or does that answer your question? Any other questions? And so one common pattern, just kind of because I think it's interesting, is you can start defining isotopic patterns in the MS1. Example for bromine, right? So you say that it has a doublet and they have the same intensity. And then you can also say, whenever you find that, make sure the MS2 associated with it has these peaks. So you can start tying across different MS levels. You piece together all these little conditions that really constrain what you're looking for. And you can combine with things like it has to have this mobility, it has to have this retention time, the isotopic pattern has to be this, the mass defect of the precursor N/Z has to be this. And so you can really start nudging little bits of knowledge, and then at the end of the day, you end up with exactly what you want. So, but the hardest part is kind of thinking about what you actually want. So that's kind of interesting. Okay, so that's about writing MassQL queries with a chatbot. But now how do we start using it in the context of some data that you've already analyzed? And so this is one of the feature-based molecular networks that I believe you guys looked at with Giovanni yesterday. And so here's a link to it. And this is the interactive network visualization. It takes a little bit to load, especially with everybody clicking on it. But if you want to click on this link, we can all go to it, and it will get visualized in a few seconds, just because it's quite the large network. And it's just interesting to me. I'm looking at the server right now. It is like, with all of you loading it, we are using like 40 cores just to load networks for everybody in this room. So that's interesting. That's fun. Okay, so now that this is loaded, so one of the things that I'll show you next is if you go to the bottom right corner, there's a way for you to highlight spectra in your network. Just so if you look at this, there's so many nodes here on the left-hand side. That's why it took so long to load, right? So there's so many nodes, and how are you supposed to start prioritizing what you want to start looking for? And so one of the ways is you could start asking the question, within my molecular network, and again, you can do this on the raw data itself, but here we're just kind of showing it interactively within the molecular network. Within the molecular network, maybe you're looking for a specific fragmentation that is indicative of certain types of compounds you're looking for. And so in this particular example, I just picked one random element out, but maybe we want to look for MS-MS spectra that have a 495.2 M over Z. And so what you can do is if you hide this, just scroll down here, there's an MS2 M over Z highlight here on the bottom right-hand side. So we do 495.2. And so this is just kind of a simple shorthand. It actually writes a MassQL query right here, and actually uses the MassQL query. But this is so that we don't have to write the whole query right now. But then once you enter that, and then this is kind of a giant network. But what will eventually show up is these purple circles. And so these large purple circles are MS-MS spectra that actually have this particular peak in them. And so that's one way that you can start really kind of tearing apart, prioritizing stuff in the molecular network if you have some sort of hypothesis of what you want to look for. And again, because we enable full MassQL expression, you can be really fancy here. You can start talking about multiple peaks, mass defect, and all this stuff, and copy and paste that MassQL query here. But here we're just showing you a simplified example. And so for one example, you can see there's this family down here in the bottom right-hand corner. Every member of that family is purple. If we want to, oh, okay, there's a, okay, I kind of screwed that one up. Anyway, but we can start looking at the fragmentation. So let's put that in here. And every member of that family seems to have this particular mass. So whether that's interesting or not, this is something I randomly chose. But this is another way that you can tease apart your networks and start really figuring out what's interesting with MassQL interactively directly in this dashboard. So does that make sense? Any questions about this interface and utility and things like that?
That's all quantities.
I'm sorry?
We can continue.
Okay, great. So, okay, so that's with MassQL. So that's kind of finishing up with MassQL. You come in with a hypothesis, you can start finding exactly what you want to find in these molecular networks. Now, especially because you all have done molecular networking in this workshop, one of the key advantages of molecular networks is you can use it to identify new compounds. And I'll just show you kind of a new tool that we just put a preprint out because we're excited about it. But one example here, this is published back in 2020. And here is a molecular network. Again, each circle here represents a molecule, and connections mean that we think they're structurally similar. And so this is work done by Robbie Quinn and Emily Gentry. And so what we can see here is, this is a molecular network. There's like 6 members. Only this one member that's large is identified. So we know this is a bile acid, this is a glycolic acid. But then there's all these other members of this family that we don't know what they are. But because they're close in the network, it gives us a hint at what the structure could be. And so what you want to be able to do is you want to say, I know this structure here, this 466 M over Z, that's glycolic acid. And there's a neighbor that is 90 Daltons heavier at 556 M over Z. What could it be? And this is kind of a natural thing in molecular networking where we can start propagating annotations from known nodes to neighbors with a similar structure. Now, the problem is, in order to propagate this, and so Emily and Robbie, they propagated this and they figured it out. It was instead of glycine on the conjugation, it was phenylalanine. But they did this manually. They had to figure out what the mass delta was and where did that structural modification occur. And it's it's faster than before in trying to figure out de novo, but it's still pretty labor intensive. You have to know a lot about the fragmentation and things like that. And especially if you have thousands of molecules that are unknown in your data, how are you supposed to kind of get through all of this, quickly, right? So, or at least faster than people are doing it right now. And so we introduced a new tool, and we'll just quickly give you the high level intuition. But it's a tool called ModiFinder. So, and the goal is to localize modification. So here we have this natural product, and so ignore the numbers for now. But what you can see here, this is our known molecule, and we just happen to observe in our molecular network that there's a methylated version of it. So we know it's methylated because it weighs 14 Daltons more, but we don't know where it's methylated. It can be on more or less any of the atoms on this molecule, right? It's a pretty big molecule. And so the natural question we want to ask is, where does the methylation happen? And so that's what ModiFinder will hopefully help you with. And so whenever you're doing these propagations in the networks, if you know a particular molecule, we can say, hey, there's a neighbor, it's modified, and the modification appears on this particular atom. And so at that point, it probably accelerates your structural elucidation work, and we think that helps. And so this is work done by Reza in my lab, and the intuition behind all of this is pretty simple. And we don't, we're computer scientists, we don't think too fancy. And the idea is, maybe you have a compound here on the top that is kind of represented by these puzzle pieces. And then you have a compound 2 here at the bottom that's very similar to compound 1, but it differs by the addition of this modification, this red puzzle piece. So let's assume we knew the ground truth. And so what happens is because when you fragment it in tandem mass spec, there are certain peaks that stay the same mass, which will not include the red puzzle piece. But there's going to be peaks that shift in mass that we know contains the puzzle piece. And so what we can do is, if it shifts in mass, we know, and then we can figure out the substructure, in this case, the green puzzle piece of the shifted peaks, then we can say, look, that new red puzzle piece is attached to the green puzzle piece in the original molecule. Does that intuition make sense? So very, very simple. The modification site has to be on the shifted peaks, and we just have to figure out the substructures of the shifted peaks. That's kind of the bottom line. Again, we won't go into so much details here, but the paper outlines kind of the computational and the benchmarking and all that stuff. And so what we produce here is, so as an example of this particular natural product, is given both of the tandem mass spectra and the known molecule, what we give out as part of ModiFinder is we tell you, hey, this is the molecule, this is the known molecule, and we give you a likelihood score for each atom across the entire known molecule. And the higher the score means there's more evidence, at least in our computational approach, that the modification happened on this specific atom. And so what we can see here, the ones in blue, we think it's very unlikely that the modification happened. And then along this right-hand portion, this ring right here, we think it was one of these carbons, 13, these the higher numbers mean higher scores. We think it's one of these carbons or nitrogen where the methylation happened. And so it narrowed it down from this whole molecule to a significant subset of the molecule. Now you have to deploy, we think this is the limits of the fragmentation in our computational method. We can't get any more specific than that, but you can use your your chemistry domain knowledge to kind of narrow it down from, instead of 50 atoms, we give you a handful, and then you have to kind of carry it to the very end. Does that make sense of what ModiFinder can help you with and give you? So let's do a little quick hands-on. So just because these tools are, I'm sorry?
Oh, sorry, I just had a question if that was okay.
Yeah, go ahead. Sorry, I didn't see it.
Yeah, sorry, I'm not on the camera. I'm a different area. So with ModiFinder, so can you upload like a custom annotation to one of the nodes and then use ModiFinder on that custom annotation, or does it need to be in one of these libraries?
So you can upload your own. So you can upload your own. We'll just show it in a second, but you just need the US size for the spectra and any structure. So you can provide your own structure. The one thing that about providing your own structure is We do actually do some pre-compute. One of the hardest steps is actually figuring out the substructures on the peaks of the tandem mass spectra. And so we happen to use MS Buddy and Sirius internally as refinement techniques. And there's also other things we do for refinement. So you can do it with your own structures. It does not need to be in a library. But if it is in the libraries that we have, we do a bunch of pre-compute and the results are better. So that's essentially the bottom line. So, but if you want to do it, or if you want to even take an existing library spectrum and change the structure, you can. So that's all. It handles it. It just won't be quite as good of results. But it's fully possible.
Ming, it's Carlos here. I had a question for you regarding these modifications. That's your approach, consider rearrangements in the molecule for
Right.
You have modifications?
Right. So this is kind of a weakness. So this becomes especially in the fragmentation steps when you're starting to annotate. I don't have a good slide here. Let me just bring up ModiFinder itself. So we can do it. We can start doing the hands-on portion and start answering these questions. But if you go to modifinder.gnps2.org, you'll get something like this. And we have a few examples up here. So, but let me just kind of sort of answer this question, Carlos. So one of the things that we do, one of the key things that we have to do is we have to annotate each peak with a substructure. Now, if there are rearrangements in the fragmentation, we don't capture that right now. That's something we're looking forward to doing in the future, because what we found is there are some peaks that we just cannot explain as a substructure of the original molecule, and we think those are good candidates for rearrangements. So we don't handle it right now. We're thinking about including those hydrogen rules from like MS Finder and things like that, but we haven't done that quite yet. But that's a good question. So we expect that to kind of improve our performance. Another thing that we did in our paper for performance kind of evaluation is we do a combinatorial fragmentation approach for annotating the MS2 peaks, and we vary the depth. So we have to figure out what is an optimal fragmentation depth, and we settled on 2. But if you have higher energies, that optimal number could be significantly higher. So those are kind of the limitations. But it's not, this annotating MS2 fragments, it turns out, is not the easiest thing in the world. So
you're right. Thank you very much.
And there was that paper that recently came out that was saying all of the MS2 peak substructure annotations and databases were all wrong. So I'm sure ours are wrong, but they're right enough to be useful. So at least that's kind of our hedge on that claim. So, but cool. Any other questions so far?
I think we can continue.
Okay, sounds great. Okay, so in terms of the hands-on, you can open ModiFinder. There are certain other examples up here just to show that things kind of work. But the key thing here is on the inputs, you have your known compound and your modified compound. So you need to have a USI to specify the MSMS of the known one. You need the USI to specify the MSMS of the modified one. And you need the SMILE structure of the known one, but you don't need the SMILE structure of the modified one. Here, we just do it for illustrative purposes. So for example, here is the prediction of the site localization. And here is the known compound with the actual location highlighted. So what ModiFinder had guessed for this particular modification where the methyl group was, it got it pretty close to being correct. There were some that were equally high score that we couldn't distinguish, but it was pretty close. And one of the reasons that it worked quite well in this particular molecule is if we zoom out, there's actually quite a bit, just to zoom in a little bit, there's quite a bit of fragmentation in this particular molecule. And there's other cases where there's very, very little fragmentation, and it's very hard to actually localize. There's just not enough information to actually localize these things. to a specific atom. But does that make sense? Any questions on this part? So one weakness that I kind of mentioned earlier is that we need to annotate the substructures for each peak. And we do it in kind of a semi-naive way, but it works, right? So, and we kind of lay that out in how we do that in the manuscript. But the thing is, these computational tools don't know as much as you about your own molecule that you're interested in. So imagine you're looking for a drug, and you're looking at all the metabolism products of the drug, and you work with this drug all the time. So you may be able to explain all the fragments of this particular drug. And so one of the things that we wanted to do is be able to have you all leverage your domain knowledge in conjunction with the computational algorithm that we introduced here. So for example, I took this peak, and we computationally think that there are two options. There's two substructures here. So it's not particularly like the most complicated thing. But one of the things that you can do is because you're a chemist and you know what you're doing, you can go in and you can say, no, okay, computer algorithm, it's not one of these two, it's actually the second, just as an example. And so if you can say that and you say it's definitely not the first one, and you click apply, one of the things that it can do is it takes your input to refine the substructures, and it will update the likelihood distribution for site localization. So this is especially useful if you want to annotate like 3 or 4 peaks with a particular substructure that you know, and then it will synthesize that knowledge of what you input in, refine all together into a unified likelihood distribution. So it's probably something if you all were solving these modification kind of challenges, something you were probably doing manually anyway, but this allows you to synthesize it in a computational framework that combines it into kind of a scoring domain, and something that's a little nice to kind of visualize and guide your analysis. So we think that's interesting to leverage both the domain knowledge as well as the computational guesses that we can start off with to help with the site localization problem. And so this is all available at GNPS 2 today, and we're building out to make this a little bit more high throughput in the future. So any questions? So these slides we already went through here. Any questions so far?
Any questions? You can continue.
Okay, so that's all I wanted to cover today in this kind of hour and a half. But overall, as a conclusion, GNPS 2, it's a new platform. So that's kind of the thing we talked at the beginning. It's a new platform that we want to enhance resiliency and access to tools, right? So lowering the barrier entry to use these computational tools. So you can take the workflows that we run there out, run it on your own systems at whatever scale, whatever computing systems that you want. If you want to run it on large scales on our side, just, you can use it on our side, but then if it's really busy and you want to collaborate, we have a pretty modest computational cluster at UC Riverside, something like 400 or 500 CPU cores. But we reserve some of those for collaborations in our own work. But also we introduced MAST and MassQL on how they could enable mining of public data at large scales, and ModiFinder as a new computational algorithm to accelerate data propagations and annotation propagations in molecular networking or analog library research for that matter. But everything is available. You should have accounts. Feel free to reach out or just try it without even talking to us. But if you have further questions, you can reach me via email. I also hold weekly office hours. I've been doing this for like the last 5 years. This quarter, for right now, it's on Tuesday mornings at 9 a.m. Pacific time. But it might shift depending on my teaching schedule every quarter. But you can just show up on Zoom. You don't need to email me. That's kind of the beauty of it. I don't need to do anything. You don't need to do anything. You just need to show up. And we have a good attendance. We have like 5 or 6 people every week. And we just talk about problems they have, implement new fixes or new features, and answer questions. So we think it's kind of a fun fun hour every week. But none of this could have been possible without, the funding sources. I've been very lucky having great collaborations as well as a global network of co-authors across all of the work we've done over the past 10 years. So I'm super thankful for that. And thank you guys for paying attention and asking great questions, being engaged. So hopefully this is useful and informative. And these are just some of the tools that we're very excited about. There's plenty more that we haven't talked about, but you can only squeeze that many into kind of a presentation and hands-on session. But also, I couldn't have done this without my lab at UC Riverside. We're humble right now. We've got we have 3 PhD students, a project scientist, and a few undergrads. But we're slowly growing. But happy to answer any last-minute questions from you all.